I am Darjat Hussain, student. Today I am going to give the lecture on the central dogma of the biology because the many students are confused what is the central dogma of the biology. So today I am clear the little concept about the central dogma of the biology. Before going to talking about the what is central dogma of the molecular biology, you have to be distinguished between what is the DNA and what is the RNA. You know the DNA is a deoxyribonucleic acid because D means removal of the oxygen from the pentose sugar is called a deoxy. That's why the DNA is called a deoxyribose, while the rRNA is known as a ribonucleic acid. In this diagram, you can see here the DNA is the double strand. You can see here this is the DNA. This is the DNA, which is consist of the long polynucleotides, which consists of the two strands. That's why the DNA is called the double X. While you can see here, this is the RNA, which is consists of the single strand. This RNA is called the messenger RNA, which is formed from the DNA. Basically, the central dogma means there's a two process included in the central dogma, in which one is the transcription and one is the translation. You have to be differentiate between the transcription and translation. Transcription means the formations of messenger RNA from the DNA. You can see here the DNA used as a template over there. This DNA help to make the single strain, which is known as the messenger RNA. This is the messenger RNA. The DNA one strain is as a template used by the different chunk of the enzymes, helicase enzymes, and the RNA polymerase enzyme. Because the DNA consists of the instructions. These instruction may be copied by the messenger RNA because DNA cannot cannot flow itself from the nucleus to the cytoplasm. That's why it needs a personal assistant, which is known as the messenger RNA. These messenger RNA copy the codes, which are complementary codes which are present in the templates of the DNA for the productions of the for the synthesis of the protein. That the template which have the 3 prime to 5 prime template used as a template strain on the against of their follow the Chergev rule. The Chergev rules, the codes are follow the Chergev rules. In the front of a governance there is a attachment of the cytosine, and in the front of adenines there is a thymines.